When Michelangelo was painting his famous painting of the creation of Adam, with the finger of God pointing to the finger of the inanimate Adam, something, we imagine, must have been passing between those fingers to animate Adam. What was it? Today we're going to talk about the possibility that if there's anything in that myth at all, it would be electricity that passed from one to the other. Because if you could make God positive and Adam in his inanimate form negative, and you brought the two together, a spark would go across. Now that's just a story. But I have another story too. When Lord Byron and Percy Shelley and his wife Mary Shelley were sheltering during a cold winter in Switzerland at Byron's chalet, they were talking about precisely such experiments in which the great scientist Galvani showed that if you pass an electric shock into a nerve attached to a frog muscle, it twitches, even though it's no longer in the frog. And if you do the same to the frog itself, it also twitches. So the idea that electricity is a key feature of life, of living systems, is something that has quite a long history in the development of our knowledge of the body. And today, Voices from Oxford is talking to one of the greatest scientists working on this question, because Frances Ashcroft is working here in Oxford in the Department of Physiology, Anatomy and Genetics. She is a Fellow of the Royal Society, which is the National Academy of Science in the United Kingdom. She has won many prizes for her work, which is on the way in which electricity passes through little protein structures in the cell membrane. But more than that, Frances Ashcroft has another skill which she has shown to delight the world, which is that she's a great writer of books for you and me. Her book, Life at the Extremes, had a huge success, and it's been followed by another book which we're going to discuss today, which is The Spark of Life. So, Fran, these proteins that carry electricity in the body, are they really everywhere in our body?
They're in every single cell that we have, and they're in every organism on Earth, from the simplest bacteria to the greatest of the redwoods in California. Every single cell has these particular proteins, and they play an extremely important role because they are the means by which ions, such as sodium and potassium and calcium, can carry electric currents into and out of our cells.
Ions, just to be clear, are charged atoms.
That's correct, yes.
So they're carrying electric charge from
and this is a very fascinating thing, because the electric currents in our body are actually rather different from the electric currents which we find running in the wires of our house. So those which run in the walls of our house are actually carried by electrons,
yes,
whereas those which are in our bodies are carried by, as you said, charged atoms.
The electron, just to be clear, is of course the little negatively charged particle that goes around the
outside of the nucleus of the atom.
And what you're saying is that what actually goes through these protein channels in the body has to be what can transfer itself through water, after all, because it has to go in a water environment, and those are charged atoms.
That's exactly right.
Now, can I ask you then the next question? If they're everywhere in the body, when you do your work on the function of these channels, my understanding is that your work has led to the understanding of some major diseases.
So I've only made a very small contribution, of course, to the field in which I'm working. Many other people have also contributed, but I'm particularly interested in understanding how the level of sugar in your blood is controlled. Right. And we know that every time you eat a meal, your blood sugar level goes up, and that it's also very important that it's kept within narrow limits, because if it falls too low, then the brain is starved of fuel and you very quickly die.
Yeah.
But if it's too high for too long, then you get the complications associated with diabetes. And the key hormone that regulates blood sugar is insulin, which is released from your pancreas in response to a rise in the blood sugar after a meal.
Right.
And so I've been trying for many, many years to understand how this process is brought about and how it's controlled. And we and others discovered a tiny protein pore that is found in the membrane of every single one of the insulin secreting cells. And when this pore is open, insulin is not released, and when it's shut, insulin is released. And what happens, of course, is that when your sugar levels rise, then the pore shuts, and that triggers a whole series of other things that results in insulin release.
Yes. Right. So there's a beautiful feedback mechanism here connected with that electric protein channel.
Yes.
Now, as I understand it, you have studied some of the ways in which those kinds of channels are changed genetically with what we call mutations.
Yes, well, there are two sorts of mutations. If one has the DNA that codes for the protein itself, you can use genetic engineering to make those mutations yourself, and thus see what each particular amino acid that makes up the protein does. So you can understand how the protein actually works by making mutations. Right. But there are also the happy accidents of nature, which mutations which are found in humans and in animals, which scientists can use to try to understand what the protein does. But of course, these happy accidents are not particularly happy for the patient.
The patient or the animal.
No, because they may lead to disease.
Yes,
and in the particular protein I work in, if you have a mutation that is overactive and doesn't shut when glucose goes up, then of course insulin is not secreted and you get diabetes. And these mutations were first found by my colleague Professor Andrew Hattersley from Exeter University, who is in fact an alumni of Oxford. And when he found the first mutation, he very excitedly called me up to tell me, and I have never ever forgotten that day. And yes, it was, because then we were able to show how they work. And what we also realized was that they could be closed. Despite the fact that they were not closed by the rise in blood sugar, they could be closed by drugs which were already in routine clinical use.
This is the sulfonylurea.
These are drugs known as sulfonylureas, which are taken by people who have adult onset diabetes. This is a quite different disease to the one that we've been working on, which is a very, very rare genetic condition where people are born with the disease.
Born with it, yes, exactly.
Yes. And in the past, it was thought that they had type 1 diabetes, which is the form of diabetes that happens in children and young adults and is caused by an autoimmune attack on the cells. That's a kind of friendly fire where the body destroys its own cells.
Attacks itself, yes.
And therefore, there are no more pancreatic beta cells and no more insulin secreting cells in these patients, and they have to take insulin all their lives. But what we realized, Andrew and I, was that because the drugs could close these channels in patients who had who were born with diabetes, it might be possible to move them from their insulin injections, which they were initially given, to sulfonylureas, and to everybody's delight it worked.
It works. It must be marvellous, Fran, to do science like that and then eventually to be able to meet the very patients that are benefiting from that science.
Oh, I think I've been extraordinarily lucky. It's very unusual for a scientist to do that.
The impact in terms of clinical practice to come so quickly.
Well, it didn't really come quickly, because from discovering that the channel was regulated by glucose to identifying the first mutation was 20 years.
Yes,
and then it was just a few years 3 or 4 years before the first patients were treated.
Right.
So
Right.
it depends from the point of view at which you look at it.
What time scale we're talking
Yes.
about, yes. Now, you've also worked on mutations of these channels in animals, and you can tell me about the falling goats, perhaps.
Oh, I have not worked on the falling goats, but I'd
Right.
be delighted to tell you.
Yes.
So these diseases in which mutations in ion channels cause human disease have been given a collective noun. They have their own collective noun. It's called channelopathy.
Right.
And the very first channelopathy was discovered by Shirley Bryant, who worked at the University of Cincinnati in the States. And it was very interesting because he was reading about a herd of goats that fell over every time the train ran past the field and sounded its whistle. And he thought, that sounds very like a human condition, which is known as myotonia congenita, in which when patients are startled or they try to do something very rapidly, they become very stiff and their muscles lock up. And for example, if you meet one of these people and you ask them to pick up a suitcase, they can pick the suitcase up, but they cannot let go because their muscle is clamped to the handle.
Yes. Yeah.
So he decided to investigate, and he set off to find some of these goats in Tennessee.
Yes.
And he immediately encountered a problem because the farmers who owned the goats couldn't believe that he wanted the mutants. So they just gave him ordinary goats. So he had to make a second trip to collect the mutant animals.
All right.
So when he got the goats back to the lab, he discovered that whereas a single nerve impulse gives rise to a single muscle twitch
Right.
in a normal goat,
Yes.
in one of the mutant goats, it gives rise to a train of electrical impulses in the muscle,
And the muscle
so that
therefore-
causes it to contract very
muscle does that,
tightly.
yes.
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 So the reason the goats fall over is because their muscles are being stimulated too much, and therefore they're contracting.
Yes.
And moreover, every time you move a muscle, you have two opposing pairs of muscles. So you have the flexors and the extensors. So when you contract the muscle like this muscle is contracting and the one on the other side is relaxing.
Doesn't it? Yes.
But in the goats, that's not what happens.
They go
Both
stiff.
of them contract,
Yes.
and therefore they go very stiff,
Right.
and then they fall over, and it looks extremely funny. So what was so interesting about the work on the goats was that it subsequently led to the identification of the gene that was defective. And
yes.
that in turn led to the understanding that human patients have similar mutations in the equivalent gene. And I think this is very interesting because it turns out to be true for a large number of mutations, that in fact one can look at what's going wrong in the animal and subsequently understand what is happening in the human.
In the human, yes.
And there are many other diseases in which this has been the case.
Yes. Now, I want to switch to another type of animal. There are animals that use electricity to shock their prey.
Ah, yes, of course.
The electric fish.
There are
The
two of
electric eel,
those. Yes,
yes.
the electric fish, the torpedo, is particularly interesting because that's been known since antiquity. And I believe it was Scribonius who suggested that the electric fish could be a therapy for gout. You had to stand on the fish
Yes.
in the salt water.
Oh my goodness.
Until
Get shocked.
until it numbed the leg and the pain was no
Yes,
longer felt. But of course some of these shocks that are produced, like the ones by the electric eel, are in fact quite substantial. They can produce a shock of 600 volts. So they kill small animals this way, and which is what they feed on. But they can deliver a pretty hefty shock to a human too.
Yes. So why don't they shock themselves?
Well, this is a mystery that nobody totally understands. One hypothesis is that most of the eel is surrounded by a fatty insulating layer which prevents them feeling the shock. But of course there must be parts where the insulation is not present because the electric current that they produce has to get out across the surface of the skin, so there can be no insulation there. So I think it's something which science still doesn't fully understand.
Yes, okay. Now I want to switch to another aspect of electricity in the body that you deal with in your marvellous book, The Spark of Life, I am a great fan of hot curry.
Hot curry?
Yes.
Uh-huh.
And of other kinds of cuisine that use the chili pepper. Mm-hmm. I can consume great quantities of the chili pepper.
Ah, you can, yes.
Now,
it's probably as a consequence of damage, Denis.
Possibly so. But now let's ask the question, what has electricity got to do with that? When I get that feeling on my tongue.
Well, the reason that chili peppers taste hot is that they have inside them a chemical known as capsaicin.
Yes.
And the hotter the pepper, the more capsaicin it has.
Yes.
And what capsaicin does is that it actually binds to and opens an ion channel in the receptors in your mouth.
Just here, yes.
Well, wherever.
Yes.
And what's so fascinating is that those receptors, those ion channels, are also activated by noxious heat. So the brain can't actually tell whether it is heat or whether it is chili pepper that is stimulating them. Yes. And it interprets both of them as heat. And that's why chili peppers taste hot.
Yes.
And very interestingly, it turns out from the work of David Julius and his colleagues in California that there is a whole range of related channels that are sensitive to a complete spectrum of temperature. So at the one end, you have the ones that are sensitive to capsaicin, which taste hot, and at the other end you have ones which are sensitive to cold temperatures, and also, interestingly, to things like menthol and eucalyptus, which is why they taste cool.
Right.
And these particular ion channels have also been taken over, if you like, and developed by animals in order to sense the body heat of their prey. So this is how rattlesnakes and pit vipers detect the their prey that's some distance away, because they have developed a very sensitive system of detecting very tiny amounts of heat.
Yes.
And it's also the way, interestingly, that vampire bats detect their prey.
My goodness. And they detect the heat of the...
They detect the body heat.
Right. So this is a little bit like the fact that if I stimulate the eye, I see flashes of light.
Yes, you mean if you press on the eye, you see presses?
Yes, that's exactly
or if,
right.
Yes.
It's as though, although you're doing something else, in this case a mechanical deformation of the receptor, what you see, of course, because the brain is interpreting it as light, and what you're saying is that my tongue is interpreting the sensation as hot or cold.
Exactly so. And of course it was Newton, as you probably know, who described this phenomenon of touching the eye and generating light first. In an extremely dangerous experiment that he did by shoving a bodkin round the back of the eyeball. Not to be encouraged.
Now I want to take you, Fran, in a slightly different direction now. In metaphor, we often talk about the excitement of interaction between people, as though their conversation is electric, their touch is electric. Does this have a connection with what you're talking about?
Well, I wonder, in one sense, of course, it does, because as I speak, your ability to hear me and your ability to see me is down to electrical signals which are being stimulated in the sensory cells that feed to the regions of the brain which are sensing sound and sight and everything else, and your ability to interpret those is down to the electrical signals that are taking place in your nerve cells. And indeed, in my view, your very personality is down to which neurons are firing at particular times. I subscribe to the view, as Percy Shelley said all those years ago, that man is a mass of electrified clay. I think it's such a marvellous poetic phrase. But I can answer your question a different way, because of course if I happen to be charged up with static electricity and I were to touch you, sparks would fly between us. And in fact, this was a party trick in the 1800s when people discovered that they could store static electricity and then discharge it. And what they would do they would for example, charge up a woman who was standing on glass slippers, so she was insulated from the floor,
yes.
And then her suitor would come along with puckered lips outstretched,
yes.
and sparks would fly between their lips. So she was known as the electrified Venus, and she gave stinging kisses.
It takes us off anyway into some very poetic questions to do with electricity in the body, and in particular you refer in your book to Percy Shelley's remark, which you also referred to earlier on in this discussion, about man being electrified clay. And what you say there is that's not too far from the truth.
Well, not in my opinion, because if you think that your personality, the person that you are in human consciousness, is down to electrical signals in our nerve cells and the chemicals transmitters that communicate between one nerve cell and another, then we basically are nothing more than soup and sparks.
Right. So that picture of Michelangelo animating Adam by pointing, the two fingers come close together.
Possibly, but I think that we should remember, of course, that it is not possible to animate a human being by merely electricity. It is possible after death to stimulate the nerve and the muscle fibers for a brief period before they themselves die. Because I think one of the things that people forget is that the death of a multicellular organism like ourselves is not an instantaneous thing. It's more of an extinction by stages, because cells die at different rates. And that's why it's possible to cause the frog's legs to twitch even though the animal is dead. And of course there were the very famous experiments carried out by Galvani's nephew Aldini, in which he took decapitated corpses or corpses straight from the hangman's noose, and put on these spectacular theatrical demonstrations where he applied electric currents to different nerve and muscle fibers and showed that the fearsome facial grimaces were produced, and that in one case the headless corpse rose up and pointed its arm to the audience, to great consternation amongst the people watching. But it doesn't happen more than if you don't get the corpse very early,
yes,
quickly.
Right.
Yes.
So, in that sense, the Frankenstein story is really a myth, isn't it?
Yes, it's certainly a myth. But of course, I think these sorts of demonstrations were one of the things that led to the Frankenstein story in the first place, and it's very interesting. Mary Shelley in her book does not, in fact, stipulate that it is electricity that animates Frankenstein. This comes out of the film in which Boris Karloff played the monster. What she says is that Dr. Frankenstein gathered the instruments of life around him. But she does infer at one point in the novel that electricity might be involved, because she describes how, as a young man, Frankenstein saw a tree struck by lightning, and he inquires of his father what was going on and is told that it's electricity.
Yes. But it's clear, isn't it, from your quote from Percy Shelley and the story of the writing of the Frankenstein novel by Mary Shelley, his wife, that their interest in the idea of electricity in the body was there in a deep way, wasn't it?
Yes, and Percy Shelley, of course, is well known for having shocked his family around the dining table. His sister describes how she really didn't like this, but he would hook them all up to a Leyden bottle which contains static electricity.
Yes. What, around the table?
Around
all
the table,
hands.
Yes.
And he'd have a Leyden jar with static electricity there.
Stored in it at one end, yes.
And then suddenly, what did they do? All jump away?
Yes, well, of course, this is a spectacular demonstration which was done a lot in the sort of late 1700s. Many people describe this. For example, 2 legions of soldiers, etc., jumped in a kind of Mexican wave, and a whole 200 Carthusian monks did the same thing. They were connected in a long circle, each holding iron bars between one another.
Yes.
And then what happens is the Leyden jar is a little bit like a capacitor. So if you connect the two ends together, the current will flow and people will get a shock. And of course, there's a very famous story about this, which you might know, which I always find quite amusing, which is that Sigurd de la Fond in around 1785 set up one of these demonstrations, and his immense puzzlement is stopped on man number 6. Man number 6, the shock was not transmitted any further. Nobody could understand this. They tried it again with the same effect, and so they went away puzzling about it, and the rumour spread round Paris that man number 6 did not have everything that constituted the distinctive nature of a man. And in other words, that you can't electrify a eunuch. And the Duc de Chartres decided that they would do the experiment. So he lined up 3 of the king's castrati and 3 entire controls and shocked them all. And of course, all the castrati leapt exactly in the same way. So it wasn't true. No. But it took some time before they realised that the reason it stopped was when somebody was standing on damp ground. Right. And what happened was, of course, that the pathway for the electric current had less resistance to flow through the person to ground.
And didn't go on to the next person.
So it's a very nice demonstration, really, of what ground in an electrical circuit is.
Right, of course. And we call it the ground now,
we call it ground, yes, or earth.
Yes, exactly. And
the point of lowest potential.
Exactly. And of course those great big bolts of lightning that come from electric clouds go to ground.
Are also shorting themselves to ground.
Yes. So just to come back finally, Fran, to the poetry and the poetic aspects of all of this, the way I see this is that the 19th century, of course we've got to remember that we're talking of days when discovery of the nature of electricity was still in its early stages. We'd still got to get to understand it more scientifically. So in many ways it was for the early 19th century people something of a bit of a mystery about the nature of life, what was really going on. So it's not too surprising that the romantic poets like Byron and Shelley and of course his wife and other poets were excited by what was emerging as early stage knowledge of parts of our understanding of life.
Yes, and of course poets are still doing this. There's a wonderful poem by Brian Turner, which is who was in fact a soldier in one of the recent wars, and he has a poem called Here Bullet, and he has this marvellous phrase, "The leap thought makes at the synaptic gap." And which describes, of course, the chemical transmitter
from one nerve to another, from one nerve to a muscle.
Yes, and then Joe Shapcott wrote a wonderful poem about different types of ion channels and the diseases that they give rise to, and has come up with another wonderful phrase, the flutter in the ion channels. Right. So poets are still writing about these things.
Yes. I like the flutter because when you and others record the opening and closing of ion channels, you actually see single channels opening and closing, single protons, yes? And sometimes they do it in a kind of flutter, don't they? You get a burst of...
Exactly so, and they're in fact called flickers or flutters.
Exactly.
And this is exactly as you say, because the channel is opening and closing, and it's possible to record them. And I think it is a wonderful technique, this technique that was invented by the German scientists Erwin Neher and Berk Sackmann, which won them the Nobel Prize, which allows you to actually record the activity of a single protein in real time. It's a magnificent achievement.
It is extraordinary. Well, Frances Ashcroft, thank you very much for talking to Voices from Oxford. I think you've convinced our viewers that they really ought to be reading your books. Thank you very much.
Thank you, Dennis.